Welcome back to another video. In this video, we're going to discuss the family tree of the immune system, all the different types of immune cells that we might find in a human body, and the way they are related to each other developmentally and functionally, and the process by which immune cells arise, develop, differentiate into all the distinct cell types that we'll learn about today is a process called hematopoiesis, which is the subject of this video, as you can see. And when you hear this word hematopoiesis, a little bit of a strange word, but maybe that the initial prefix there, heme, might make you think of something like hemoglobin. And that's because we are talking about something that's related to blood. And in fact, we associate immune cells very heavily with the blood. Blood is a major source of immune cells in the body. It's where most of them are found, more or less. And you may have seen in other contexts that blood has several constituent parts. So, for example, if you centrifuge blood to sort of isolate the cells in the blood away from the liquid portion of the blood or the plasma, you might get something that looks like this, where about 55% of the total volume of a tube of whole blood is plasma containing mostly water as well as like small soluble molecules. And then another major chunk of that, 45%, are going to be what we call erythrocytes, or red blood cells, those cells that carry oxygen around the body and do sort of that most classic function of blood that we think of. However, between the plasma and the erythrocyte fractions, we will derive something that we refer to as a buffy coat, which actually represents less than 1% of the total volume of blood. But within that tiny little fraction of the whole blood are all of the cells that we will really be focused on this semester in terms of the immune system. This is a population of cells called leukocytes. Leuko is a prefix meaning white. These are white blood cells, which distinguishes them from the red blood cells or erythrocytes. And so you can see a list below leukocytes there of several of the cell types that we'll talk about today as we talk about immune cells, leukocytes. So anytime you see leukocytes as a terminology, it means white blood cells, and generally you can think of it as referring to specifically immune cells that circulate in the blood or derive from the blood one way or another. However, immune cells do not start their life in the blood. In fact, all leukocytes are developmentally derived from the bone marrow. And hematopoiesis, or the development of blood cells, is something that is very intrinsically tied to the bone marrow. It's where it occurs. And within the bone marrow, we find a rich population of cells known as hematopoietic stem cells. This stem cell population is capable of differentiating into many, many different types of daughter cells, which hopefully you can see in the diagram here. We are vastly oversimplifying this process on this slide. I sort of want to put a little warning there. That's mostly for the purposes of understanding in a survey course like this. There's a lot of intermediate progenitor cells and steps along the way. But I think for the purposes of a first pass at understanding the immune cell family tree, as it were, we can think of hematopoietic stem cells giving rise to two major sort of branches in the immune cell family tree in the bone marrow. Those are myeloid cells, which you can see derive from an intermediate progenitor cell called a myeloid progenitor cell. And then a lymphoid side of the tree, which derive from an intermediate progenitor known as a lymphoid progenitor. And we will see that there will be exceptions to what I'm about to say, but generally, as the most basic rule of thumb, you can sort of think of myeloid cells generally as being more associated with innate immunity, and lymphoid cells generally being associated with adaptive immunity, although we'll put some asterisks on that later in the video. But for now, let's just kind of look at each side of the family tree just a bit. You can see that there are many types of myeloid cells that descend from myeloid progenitors. They include things like eosinophils, basophils, so on. You can see the list here. They also include a type of cell called a megakaryocyte, which is important for making platelets. Megakaryocytes and platelets are developmentally related to immune cells, but they themselves are not really thought of as classic immune cells. I want to put them here just so you sort of understand that they sort of come developmentally from a similar source. But it's really all the other cells that you see here on the slide that we think of more classically as immune cells, leukocytes of the immune system. Um, so what we're going to do in this video is just kind of start and sort of break down each of these two halves of the family tree, the myeloid side and the lymphoid side, into some subdivisions or subfamilies within those categories, and then really kind of take a step-by-step, cell type by cell type sort of tour of the different major populations of immune cells in the blood and what their major functions are and sort of how they're related to each other. So let's start that journey looking at myeloid cells, which again derive from myeloid progenitors in the bone marrow. Myeloid progenitors themselves can further differentiate into two broad classes of myeloid cells. These are sort of subfamilies that fall under the myeloid umbrella. These are cells called granulocytes, which get their name from the granular appearance that they have under the microscope. And even in these cartoons, you can see they sort of are spotted. They have all these little dots inside their cytoplasm, which are cytoplasmic granules, which contain some very interesting cargo, which we'll discuss as we get into each cell type individually. But sort of as a family, this appearance, this physical appearance, sort of gives them this name, granulocyte. The other sort of subfamily under the myeloid family tree are cells that we call mononuclear phagocytes. And you can notice that this includes a cell type called a monocyte, and monocytes themselves can further differentiate into two major cell types, dendritic cells and macrophages. However, let's just think about these cells as a family for now, and think about the name mononuclear phagocytes. Phagocytosis, hopefully by now, you can recognize these are cells that are very good at eating things, and that's very important for their function, as we'll see. However, this name mononuclear refers to the idea that they physically appear to have a single nucleus. And that probably might not sound very surprising when you think of a cell in general. Most cells only have a single nucleus. However, in the context of myeloid cells or immune cells generally, you may notice that some of the granulocytes have some kind of funky looking nuclei, neutrophils, eosinophils, basophils especially. They can sort of have the appearance of multi-lobed or even sometimes even having more than one nucleus. And so the other side of the family tree that have more normal appearing nuclei we call mononuclear as a way to distinguish them from granulocytes. So what we're going to do is sort of keep that we have these two sort of subfamilies of myeloid cells in mind, but let's first sort of walk through the different types of granulocytes and see the types of functions that they perform, particularly within an innate immune response. So the first major type of granulocyte that we'll discuss is a cell called a neutrophil. And I have a few key bullet points here that sort of summarize some major facts that I think are important to keep in mind about them and how they function. One major fact about neutrophil is that they are actually the most abundant leukocyte in the blood, and in fact they represent somewhere between 50 and 70% of all the white blood cells in the circulation. So a very, very common immune cell within a given volume of blood. Related to something that I just said about the appearance of their nuclei, they also have an alternative name called polymorphonuclear leukocytes, or PMNs. You'll see that abbreviation frequently referring to neutrophils. And from this name, poly meaning many, morphonuclear sort of meaning the shape of the nucleus, they sort of appear to have multiple nuclei. So they are sometimes referred to as being polymorphonuclear. Neutrophils in particular are very short-lived cells. They are born, they sort of do their function, and they die very quickly. And that sort of, live fast, work fast sort of persona that they have sort of matches their status as the initial first responders to infection. So neutrophils are recruited to the site of an infection or an injury almost instantaneously. They are basically the first cell type on the scene to respond to whatever is going on. And they do respond, and then very quickly they die and are turned over. The things that they're doing when they're recruited to the site of infection include phagocytosis. In fact, they are very good phagocytes, and they are particularly good at engulfing and killing bacteria and fungi that are found within some sort of infected tissue. And so they can not only use phagocytosis to kill these pathogens, but they also have the specialized function whereby they exude nuclear material, part of their own nucleus, in something called a neutrophil extracellular trap, or NET. If you don't know, nucleic acid, DNA in particular, is very sticky. And by exuding these kinds of strings of DNA into the environment, this actually physically traps pathogens in the extracellular environment. They sort of get caught up in these strings of DNA. And the NET, the neutrophil extracellular trap, also contains a lot of other antimicrobial and cytotoxic effector molecules, which then are sort of able to bind very effectively to those trapped pathogens and kill them. So neutrophils are very, very effective cytotoxic effector cells of the innate immune system. I want to highlight that. We saw, for example, a type of adaptive immune cell, a CD8 T cell, which was very good at cytotoxicity. CD8 T cells are doing that through the recognition of antigen. Neutrophils do not recognize antigen. They are not adaptive immune cells. They recognize non-self primarily through pattern recognition, through the activity of pattern recognition receptors, which is going to initiate many of these effector functions that we've listed here. So these are a few key facts about neutrophils. If we sort of move down the line within the broader family of granulocytes, those cells that have all these cytoplasmic granules that they can release in order to exert some antimicrobial function. Another one are eosinophils. And eosinophils get their name from their appearance under the microscope, wherein if you do an eosin stain, they tend to turn bright red and orange. And so the histological nature of these cells is the origin of their name. And as you can see, they sort of have a bilobed nucleus, and depending on how you're looking at it, sometimes it can even look like two distinct nuclei. Eosinophils, we will not really spend a ton of time on this semester, and one reason for that is because they are pretty specialized against a very particular type of infection, and that is with large multicellular parasites, or helminths, worms, basically. So these really large helminth infections, we use eosinophils to guard against. However, as it turns out, in the industrialized world, most of us are not really commonly getting worm infections anymore. This was very common throughout evolution, throughout history, as you can imagine, and still in some parts of the world, helminth infections are common. But in most of the so-called developed world now, helminth infections are relatively rare. But where we actually see eosinophils being involved is that they are actually major effectors of allergy and things like asthma, autoimmune disorders. And so we're going to learn much later in the semester about ways that allergy and some types of autoimmunity actually arise from the types of immune responses that we normally use to fight helminth or worm infections. So stay tuned for that. A lot of interesting biology there. But since we're still at the beginning, what I primarily want you to associate eosinophils with are granulocytes that are very good at sort of secreting molecules from their granules that are specialized for attacking helminth infections. Basophils actually are pretty similar to eosinophils, and basophils themselves actually have a related cell type called a mast cell. And so I'm actually putting them here on the slide together because they have quite a few things in common. So what do basophils and mast cells both sort of do? They both contain large granules which are specifically rich in histamine, heparin, and other types of proteases. You've probably taken an antihistamine in your life, and you can sort of imagine some of the things that histamine does if that's the case. Very important for regulating things like vasodilation, vasoconstriction, airway constriction, and that type of thing. And so that's one of the reasons that it's related to allergies. These are some of the things that happen during allergy, but these are also, again, as I said, important for fighting parasitic infections as well. And this is accomplished both by basophils and mast cells. These are both major sources of histamine and other types of regulatory molecules like heparin in the body. However, basophils and mast cells are distinct cell types. Basophils are relatively rare in the blood, and they are very short-lived. We find them only in the circulation primarily, where they represent less than 1% of all the white blood cells in the blood. Like all immune cells, they develop in the bone marrow, but they spend their life in the blood and respond to helminth infections and other parasite infections specifically that we find in the bloodstream. In contrast, mast cells are very long-lived tissue-resident cells. You don't find mast cells in the blood. Instead, they migrate into the peripheral tissues, the skin, the muscle, the gut, so on and so forth, all these other sort of outside of the bloodstream tissues, where they take up long-lasting residence. And they will respond to parasitic infections as well as sort of allergic stimuli within those tissue compartments. And so they have very similar functions, but they primarily differ by the length of their life as well as the tissue compartment that we find them in, the blood versus the sort of extravascular tissue. Okay, so that includes, or that, that set of cells, neutrophils, eosinophils, basophils, and mast cells. These were our granulocytes. So let's move on now to that other sort of subfamily of myeloid cells, which are the mononuclear phagocytes, which you can see here. They include, again, monocytes, macrophages, and dendritic cells. And the things that all three of these cell types share in common is that they have a single, non-segmented, normal-appearing nucleus, hence the name mononuclear cells. All of them are very high in phagocytic capacity. They're very good at eating things. And in fact, the mononuclear phagocytes are all professional antigen-presenting cells. They both express MHC class I and MHC class II. And this is another thing that distinguishes them from granulocytes. Granulocytes are not professional APCs, whereas the mononuclear phagocytes are. They are very, very good antigen-presenting cells, and the expression of MHC class II is one reason for that. However, we have three different cell types here, so let's try to distinguish monocytes, macrophages, and dendritic cells from each other. Monocytes are circulating precursors. They are short-lived cells that we only find in the blood. So they were similar to basophils in that respect. We do not really find them in tissue. They are in the circulation. However, when we have an infection or some kind of injury, they are rapidly recruited out of the blood into the infected or injured tissue. And once they move into the tissue, that is a strong developmental signal to them to differentiate into macrophages or dendritic cells. So you may remember that the chart that we looked at originally, macrophages and dendritic cells both sort of derived from monocytes. There was a little bit of a sub-family tree going on there. That's what happens. Monocytes come out of the blood, and once they enter a tissue, they become one of these other cell types. Macrophages, one of the things that monocytes can become, are long-lived tissue-resident phagocytes. So again, once they are differentiated from monocytes within a tissue, they tend to stay there for quite a long time. And phagocytosing not only pathogens when you have an infection, but they also are usually important for things like phagocytosing dead cells or debris and sort of maintaining tissue homeostasis. And in fact, macrophages are not only important for an inflammatory response and immune reaction, but are also very important for the resolution of inflammation and wound healing, as we, again, will see a few examples of as we go through the course this semester. Separate from macrophages, however, monocytes can also become a cell type known as a dendritic cell. And dendritic cells are really highly specialized professional antigen-presenting cells. So while all three of these cell types are professional antigen-presenting cells, dendritic cells sort of take it up a notch. They are even more specialized, have even more characteristics that make them very effective antigen-presenting cells. And they actually can live in tissues, but they then can migrate around the body, especially to other types of tissues called secondary lymphoid tissues, which we'll discuss in the next lecture. But for now, basically, a secondary lymphoid tissue is some organ in the body which is rich in T cells in particular. And so by migrating to those tissues, they're able to interact with T cells and activate them, facilitating antigen presentation. And so this is a unique function that they serve, which is absolutely critical for priming, as we say, an adaptive immune response. So that finishes our discussion of myeloid cells. And just to reiterate, we had myeloid cells, within that granulocytes and mononuclear phagocytes, and then we just kind of talked about each of the cell types involved in that. Now let's shift our attention now to the other major type of leukocyte that descends from hematopoietic stem cells. And these are cells of the lymphoid lineage, those that descend from lymphoid progenitor cells. Another name for basically all of these cell types are lymphocytes. So any cell that descends from a lymphoid progenitor we refer to as a lymphocyte. This is another word that you will see very commonly, and it's really important to be able to distinguish this word from a very similar sounding word called leukocyte, which we saw early in this lecture. So remember that leukocytes are all white blood cells. So basically all immune cells are leukocytes. But only those leukocytes which descend from lymphoid progenitors are referred to as lymphocytes. So you can sort of think of the Venn diagram of which cells belong to either of those two names. But looking at the lymphocytes that you can see here on the slide, we have some cells that we've talked a bit about already, T cells, as well as B cells, and a new cell type called an NK cell, or a natural killer cell. And then you can also see that B cells can differentiate into a further cell type called a plasma cell. So let's talk about each of these lymphocytes and their sort of unique properties. So I said before that in general you could think of lymphoid cells as adaptive immune cells, but natural killer cells are the major exception to that rule, and that's why we're going to talk about them first, because they're a little bit different. Natural killer cells are lymphocytes by developmental ontogeny. They come from lymphoid progenitors, but they are fundamentally innate immune cells. They do not mediate adaptive immunity, and that's because they do not recognize antigen. They do not have antigen receptors. In fact, they sort of do the opposite of an antigen receptor, whereas as opposed to recognizing MHC, they can actually detect the absence of MHC on an infected or a cancerous cell of some kind. And this is very helpful. This is something that has arisen through evolution because viruses and in fact transformed cancer cells have found it very advantageous to accumulate mutations that actually turn down the expression of MHC on the surface of the infected or the transformed cell, because without MHC on their surface, T cells can't recognize them, and that's a way to survive and evade the immune response. So as a sort of counter-evolutionary measure, we've developed these cells called natural killer cells, where when they come into contact with a cell that does not express MHC class 1, that's usually a pretty good sign that it is infected or oncogenically transformed or otherwise mutated or damaged somehow, and therefore needs to be targeted for killing. And so natural killer cells are very important cells for not only for pathogen control, but actually for the process of immune surveillance during cancer formation for that reason. And as you can see here, natural killer cells also contain granules. However, we do not call them granulocytes. Again, granulocytes is a term reserved for that population of myeloid cells that contain granules. Natural killer cells are just sort of weird cells. We find them sort of in a weird place on the family tree. They do have granules, and they do release those granules, which are full of cytotoxic molecules that sort of kill the things that the natural killer cell is coming into contact with. However, they do not do that through the detection of antigen like the other lymphocytes that we're going to cover in the next few slides. Okay, so these are natural killer cells, or NK cells. Let's move on to more traditional types of lymphocytes, and let's start talking about B cells and a cell type that derives from B cells called plasma cells. So B cells detect antigen, and we haven't really looked at them as closely as we have T cells yet. But just like T cells are able to detect antigen using an antigen receptor, B cells also have antigen receptors called BCRs, or B cell receptors. And the BCR on the surface of a B cell allows it to interact with antigen, and when a B cell is activated, it is going to produce antibody. That is its primary effector function. So antibody secretion from B cells, these antibodies are going to bind to pathogens and sort of prevent their ability to interact with their targets, and then antibodies themselves tend to recruit other types of effector molecules and effector cells, which will then attack those pathogens that are coated with antibodies. We'll say much more about this in lectures to come, but for right now, you should really just be associating antibody production with B cells. And another major point about B cells, which may be a little bit surprising, is that they are actually also professional APCs. Even though they are lymphocytes and they themselves recognize antigen, they also express MHC class II, and therefore not only can they recognize antigen, they also present antigen specifically to T helper cells, CD4 T cells. And as we'll learn, this crosstalk between B cells and CD4 T cells is actually critically important for generating B cell responses and antibody production during an infection. However, for now, let's just focus on the basics, and we should also remember that B cells, after they are activated, can differentiate into a new cell type called a plasma cell. And plasma cells really sort of turn down all the other B cell functions that a B cell can have and put all of their metabolic resources, all of their cellular machinery into antibody production. They sort of become antibody factories. And in fact, oftentimes they migrate to the bone marrow where they establish extremely long-lived residency. In fact, they can live for years and even decades in the bone marrow, constantly producing antibody, which is a source of immunological memory, a way that we are sort of, we are conferred long-term protection against certain types of pathogens is because we have very long-lived plasma cells making antibodies against those infections. So it's important to say that because plasma cells put all of their energy into making antibody, they no longer do things like present antigen. They lose all of their other B cell functions, and they really are just convert into full-time antibody factories. But they are very, very important in fulfilling that role. Okay, these are just some basics about B cells. Let's now move on and talk about T cells. We've talked about T cells a bit. I'll try not to repeat myself too much, especially after the last lecture. But hopefully you remember that T cells also recognize antigen, and they recognize antigen through something called a TCR, a T cell receptor. But T cell receptors are unable to interact directly with antigen, as we've discussed, and they require a presentation of that antigen on molecules called MHC. And these MHC molecules interact with the TCR with the help of two co-receptors, CD8 and CD4. And if a T cell expresses the CD8 co-receptor, they tend to be directly cytotoxic, whereas if they express the CD4 co-receptor, we refer to them as T helper cells, wherein they modulate, or their function then is to modulate the activity of other immune cells. And that primarily occurs through the secretion of signaling molecules called cytokines. So let's summarize this whirlwind tour of the immune cell family tree. We started by saying that immune cells begin life as hematopoietic stem cells, or HSCs. This is the stem cell population from which all immune cells descend. And HSCs can further subdivide or differentiate into two major lineages. Those include myeloid cells, under which we saw that there were two designations, two subfamilies, mononuclear phagocytes and granulocytes, or HSCs can become lymphoid cells, and that as a subdesignation we refer to as lymphocytes. Immune cell families have common functions, as hopefully was clear in this lecture. Myeloid cells are generally phagocytic, generally promote innate immunity, whereas lymphoid cells are generally antigen-specific and promote adaptive immunity. Although, of course, we at length discussed how NK cells were an exception to that rule within the lymphoid lineage. Finally, immune cell identity and function can change over time and tissue microenvironment. So we saw that some immune cells are only found in the blood, some are only found in tissue, some start their life in the blood and then they move into the tissue, and then that changes their identity, it changes their function somehow. And this is a theme that we'll see develop over the course as well. But it's another major takeaway I'd like you to pull out of this discussion of different types of immune cells. And with that, that will finish our discussion of hematopoiesis.